Bacillus anthracis. Bacillus anthracis has its own importance as a bioweapon. So it is necessary to discuss Bacillus anthracis as a bioweapon or the biosafety measures that we have to take in the labs by handling this type of pathogen. Now, what is Bacillus anthracis? Here we will talk about its morphology, about its genomics, and also about treatment and the biosafety levels that we have to follow while handling these pathogens. Keep in mind that Bacillus anthrax is a causative agent of anthrax. As you have heard the name of anthrax, as these are the spores that can be transmitted from one area to another, and it can prove to be deadly. Now, this is a common disease of livestock and it can occur occasionally in humans. If we look at the morphology of Bacillus anthrax, then keep in mind that it's a gram-positive spore-forming bacteria. Usually on gram staining technique, we differentiate bacteria into two groups. One is gram-positive and the second one is gram-negative. Bacillus anthracis is a gram-positive bacteria as it has a thick layer of peptidoglycan and a thin layer of lipid. That's why it retained the crystal violet dye even after decolorization. Now, this gram-positive rod-shaped bacteria is endospore former. और अगर हम इसके एक्चुअल साइज की बात करें, then it is 1 to 1.2 millimicrons in width and 3 to 5 millimicrons in length. It can grow on a simple nutrient agar under both aerobic and anaerobic conditions. Now, Bacillus anthracis, either it is on the basis of their genotype, that is on the basis of their genetic makeup, or on the basis of their phenotype, that is the physical appearance of a pathogen. They resemble to two types of bacillus. These are Bacillus cereus and Bacillus thuringiensis. Now, these three species share morphological as well as cellular dimensions with each other. Now, if we talk about the endospores of Bacillus anthracis, they are oval in shape, located centrally in the unswollen sporangium. And these spores allow the bacteria to survive them under harsh conditions, that is in the form of high temperature, in the presence of chemicals, or when there is nutrient content is very low. So because of these spores, the bacteria can survive for decades and even for centuries in such environment. Now, if we look at the spores of Bacillus anthracis under microscope after performing spore staining, then spores are refractile and they have adipoclonic acid. If we look the spores under electron microscope, then here they have a thin outer spore coat, thick spore cortex, and the inner spore membrane surround the spore content. Now let's look at the genomics of Bacillus anthracis. It's a single chromosome because it is a bacteria, and you can see the size of this DNA molecule in base pairs. Now keep in mind that this bacteria has two circular double-stranded DNA plasmids that are labeled as PXO1. Here small p is representing the plasmid and the second plasmid is PXO2. Now, both of these plasmids helps in the virulence and in the pathogenicity of this pathogen. But keep in mind that these plasmids belong to distinct families. Now, PXO1 encode the anthrax toxins. These are PAG, commonly known as PA, known as protective antigen, LEF or simple LF, that stands for lethal factor, and the third toxin component is the SIA component. Now PX2O2, that is the second plasmid found in the genome of the Bacillus anthracis. It encodes a gene, or it has an operon that stands for CAB BCADE, The purpose of this plasmid is to synthesize polyglutamate capsule, and the importance of this capsule is that it allows the bacteria to invade the host immune system and protect them against the process of phagocytosis. I hope you people know that phagocytosis is the process of engulfment by the immune cells. So this capsule of Bacillus anthracis protects the bacteria from the process of phagocytosis. Now let's look at the Bacillus anthracis lifespan or life cycle. Most time the Bacillus anthracis spores are found in the soil. They don't perform DNA replication at this stage, so they are in the dormant stage, and due to this reason evolutionary rate of Bacillus anthracis reduces, or in other words they lack diversity among species or among subspecies level because of their short evolutionary history. So the capsule plays a very important role in the virulence of the bacteria and also shows the antiphagocytic activity. Keeping in mind this, we also talk about the plasmids that encode exotoxins. Exotoxins are actually the extracellular antigens, and it has three factors: edema factors, lethal factors, and we will also talk about the proteins. Now let's talk about the edema factor. This is actually a calmodulin dependent adenylate cyclase that increases the level of intracellular CAMP, as a result of which severe swelling or edema occurs. The second toxin is known as lethal toxin, which is responsible for tissue necrosis, means the tissue is comprising of dead cells. इसके अलावा we will talk about the special proteins that are the protective mediates and allow the pathogen to enter into the immune cell via these lethal and edema toxins. Now Bacillus anthracis can affect the skin, it can affect the lungs, heart, and even the gastrointestinal tract. Now number of vaccines are available for human beings and also for the preventive livestock use. It can be easily treated with an antibiotic penicillin, but after that they become resistant to penicillin. Now we use ciprofloxacin or doxycycline for the treatment of Bacillus anthracis. Now the spores of Bacillus anthracis, as you have seen, that they can remain in the dormant stage for many years, and these spores allow them to survive under harsh conditions. That's why they survive for decades and even for centuries. The spores can be used as biological weapons, और अगर हम history देखें तो USA, Japan, UK, Russia and even Iraq has used the spores of Bacillus anthracis as a bioweapon.